Next up, I'm thrilled that we have Dr. Heather Pua, who's going to tell us about her background and a story about her work as well. Thank you so much, Heather, for being here.
Excellent. Well, it's my pleasure to talk with you all today sort of about my journey. I'm at a little bit different stage in my professional path than Dr. Hu, so that's great. It gives you sort of a different perspective as I'm a relatively still not new, but still junior faculty member here. So I broke the talk up into two parts, sort of the first part about my personal story or my history, and the second part about research. So who am I? My name is Heather Pua. I'm a physician scientist here at Vanderbilt. Right now, I'm an assistant professor of pathology, microbiology, and immunology. And I think I have what's sort of considered the classic split for a physician scientist, which means that I spend about 80% of my time running my own basic science research lab. I think sometimes we're having a bit of identity crisis because we're doing a lot of things right now, but it makes it a lot of fun. So my roots are in immunology, and you'll see that, but we do RNA biology, and we also have worked on extracellular vesicles and are getting more and more interested in that area. I've been a faculty member for almost 4 years now at Vanderbilt, and sometimes it's even hard for me to fathom that and to look around in my lab and realize that, wow, I have 5 people in my lab now. Doesn't feel that long ago that I was still a postdoc myself. So you can make it down the yellow brick road to the end goal. It can happen, and it's pretty thrilling when it does. I also spend 20% of my time doing clinical service. I'm a pathologist by training, and my service is in the molecular genetic pathology lab. And so I basically sign out and help develop testing for genetic diseases. So those are inherited genetic diseases, as well as all the genetic profiling that's going on in tumor samples. And so I'll talk a little bit later sort of how I made decisions to go down that avenue kind of in the story. So I guess one of the questions that was asked, Beth prompted us with is to think about what your earliest influences in STEM might be. And for me, this is probably 2 teachers. The first teacher was actually my mother. So she was a high school chemistry teacher, and definitely my first example of a woman in science. And so it was very normal and natural that science would be a part of one's life, because I had this great role model at home, somebody who had done science in college and gone on to basically spend her entire life doing science, in her case, teaching. And so she was really kind of, I think, my first model of a scientist, especially a female scientist. My second early influence in STEM actually came in high school in my AP biology. And I had a really wonderful teacher, and I think it was really the first time that my imagination was totally captured. Everything we learned, I found fascinating. Trying to understand the way that cells and molecules work was elegant and intricate and thrilling to me. So I think those were really what got me interested in STEM. And even by the time I went to college, I was really probably already very committed to being a scientist in some form. So because I knew that I was really very interested in science, I looked for opportunities to do undergraduate research and started doing it very early after my freshman year in the summer with a summer program, which was instrumental, I think, in getting me started in a lab. I continued with that lab throughout my time as an undergraduate. And there I was working in a lab that worked on Drosophila. And actually, I really loved the work there. I loved working with the flies. We worked on a protein called Peewee, whose function really was not understood at all at that time, but was known to be important for germline stem cell maintenance. And so it was kind of early days in that field, but we got to do lots of interesting experiments, crossing flies, making genetic mutants, doing some molecular biology, doing some dissection of flies. And I pretty much, you couldn't kind of keep me away from lab. Like I always wanted to go into lab. And from this experience, I kind of already knew that I was passionate about science. I was always happy to go to lab. Nobody had to force me. I never had to drag myself out of my dorm room to do that. I was really lucky to have a wonderful mentor as well, who was endlessly enthusiastic and positive with me, wanted to talk to me even as an undergraduate, and paired me with senior scientists in the lab who really taught me a lot. And so I sort of was on this train where I was going towards, I think, graduate school probably. And then in my junior year of college, I had a number of things that maybe refocused me a little more towards the MD PhD. So I both had a couple of different advisors start talking to me and saying, you know, would you consider doing an MD PhD? And I said, what is that? I don't even know what that is. And I didn't have anybody in my close family who were physicians. So I didn't, you know, I'd gone to doctors, but I didn't honestly even consider it before that time. But I had by sort of another happenstance, this interesting experience volunteering at the Red Cross over the course of college, which I had just gotten involved with because of some friends I had in my dorm who were really involved in it, and they sort of pulled me along. But I got really interested in it, and I didn't actually do any of the stuff involved with the blood banking, which is maybe what you would think sort of medically related. I was actually part of their disaster services group, and I ended up leading my own disaster services group that would go out. And what we would typically do is, especially after fires, we would go and visit the families and see what services we could provide them to help them get through that period where they had just lost everything. And it doesn't sound like a classic clinical experience, and it definitely is. And I wasn't shadowing a doctor in the hospital, but dealing with people in crisis and being able to actionably do something to help them, I think had kind of a strong impact. And so I started to think both on an intellectual level that I really would like to do research that was basic but had implications in medicine, and then also kind of exploring and finding this new side of myself that maybe that maybe medicine could also fulfill. So I ended up applying for MD PhD programs. I didn't take any time off. I'd had a lot of research experience, so I was pretty confident that this is what I wanted to do, and I went straight into my MD PhD program. So my MD PhD program is actually set up very similar to Vanderbilt's now, where we do 2 years of medical school, then we go to graduate school, and then 1 year of medical school at the end. So the first 2 years of medical school was really immersive. We do classes in our first year, and then clerkships, basically doing rotations in all the basic clinical disciplines in our second year. So I had a pretty good sort of sense of what medicine was and all the different fields by the time that I went to lab. And I had worked, I had worked under Drosophila. I basically worked in a development lab, and I liked that a lot. But the thing that really captured my interest and imagination, especially starting in classes, was immunology. And I distinctly remember in medical school that I was probably the most excited when we had our immunology block in medical school. And over the subsequent year, and then throughout my clerkships, I think what I saw over and over again is that we have this complex understanding of immunology, and that immunology probably touches every single other aspect of the body and medicine. And so that to me was really interesting intellectually and very attractive in terms of application. So I went ahead and I joined the immunology program as a graduate student. And I did my thesis on the role of autophagy, which also was just very new, kind of emerging field. People basically didn't know what autophagy was until a few years before I started my graduate work, and we had the first genetic tools. And so I was able to kind of explore this unknown territory, looking at the role of autophagy in T cell survival and mitochondrial clearance. And this was after, let me just say, the first project that I did in the lab for the first year, totally failed. So we switched projects at that point, and I started up this new one, and it was great. It was productive, it was interesting and exciting. Towards the end of that, I had to kind of make a decision about what medical specialty I wanted to be in. I was obviously committed to immunology as a discipline, and so I was trying to weigh back and forth between rheumatology as a discipline of medicine, since that is largely a study of the immune system and autoimmunity in particular, versus pathology. And we hadn't had a lot of exposure to pathology, but I absolutely adored my pathologist teachers in medical school during the first year. And so I went ahead to explore that more clinically. And it was a tough decision in the end, but I think that there were two things that really ended up drawing me to pathology, and why I made that choice. The first one was somebody gave me the advice to think about what type of busy work you least detest. Because a lot of medicine, there is a lot of busy work in medicine, and there's repetition in medicine. I did not particularly like pharmacology. So I like the concept of pharmacology, but like the practice of dosing patients with different doses and changing medicines was not so interesting to me. But pathology let my busy work be looking at slides or assays, thinking about the implications, and then describing and synthesizing them, and always thinking at the level of cells and molecules. And that I can do endlessly. So I basically decided to become a pathologist. The other thing I think would be a recommendation about sort of your intuition is as you do these things, if you go in to an MD PhD program, is as you're doing that, think about who you really enjoy being around. And that often is a good indication of the specialty you should end up in. And once again, like when I was doing pathology as a medical student, I just really enjoyed going to work every day. I really liked all the pathologists I met. It's a different group of people. So most people don't go to medical school to be pathologists since we don't have patient, generally we're not patient facing and things like that. But I felt like I had found my home. So we packed up at that point, headed out in our car westward ho to do the next phase of training. And, this next phase was sort of residency and fellowship and then postdoc. And, I basically spent about 3.5 years doing clinical training at the beginning of this, doing my pathology training. There's complexities in pathology training. I won't go into that because you guys are far from that, but definitely if you're interested in pathology, make sure you talk to pathologists because there's some complexity there. But, I decided that really I had this great interest in understanding molecules and how molecules basically affect disease. And so I picked molecular genetic pathology as my specialty because I really felt like it was the part of pathology that was most close to that intersection between science and medicine. It's basically like practicing translational medicine always. So I picked that as my specialty. Finished that up, and then I headed to lab. And for my postdoc, I still wanted to do immunology, but I wanted to get more molecular. I'd really worked more on an organismal and cellular level. So I picked a lab that studied immunology, but studied RNA biology within that context. And that sort of then led me to kind of getting fellowships and things like that and being competitive to apply for jobs. So we did those 8 years out on the West Coast, and then we packed everything up. We had a few more things that came with us this time, 2 kids at that point, and we drove back across the country and came here to Vanderbilt. So what does my lab do now? Well, as I mentioned, sort of we're doing a lot of things, but I think conceptually I can really organize what the thinking of our lab into 2 branches. Really, we're interested in non-coding RNAs and both their ability to regulate effector cell programs, so basically the functional programs of immune cells, and then how we leverage that to understand or discover something about the pathways in those cells that really dictate their behavior. And we're most interested in my lab in chronic inflammatory diseases. We don't usually study infectious etiologies, but we study diseases where you have chronic sterile inflammation. And what you're going to see is that this was sort of where I started. And then as I ended my postdoc and have transitioned into my own faculty position, I've actually gotten interested in some of these cell extrinsic functions of these RNAs. And their packaging, which is in extracellular vesicles, and how this may actually contribute to chronic inflammatory diseases. Okay, so that's sort of the personal story. I'm doing okay. Next I'll tell you a bit about sort of the project that ended my postdoc and kind of bridged me into my faculty position. And I'll tell you about some of the bumps and questions along the way for that. So this project was initially rooted in thinking about asthma, which is the most common chronic lung disease worldwide. It affects more than 200 million people, so it's incredibly prevalent. And on the right-hand side, you can see a little meeples plot that shows the prevalence of asthma in the US, which is above 8% in the population. Clinically, asthma is defined by reversible airflow obstruction, bronchial hyperresponsiveness, airway inflammation, and airway remodeling, but it's truly a very heterogeneous disease. Largely what I've focused on so far is what's called the Th2 high endotype, and this is basically a subset of disease that's characterized by allergic inflammation. So that's atopy, eosinophils, and IL-13. And this allergic inflammation in the lung is really robust and responds to a broad range of allergens. And honestly, probably everybody in this audience knows somebody who has asthma. It's so common. But this inflammation is really driven in large part by Th2 cells. So this is a flavor of T cell that can develop in the body. But it leads to huge changes within tissue. So on the left-hand side, you see the normal airway. So this is a bronchus, and this is a conducting space for air to come into the lungs. And then you can see all these little sac-like structures around the outside, which are the alveoli. In a patient with severe asthma, there are many changes in the lung that are really driven by this chronic, unending inflammation. Here you can see the epithelial cells are stained in this kind of purpley violet color, and this is actually a stain for mucin. So they produce abundant amount of mucus secretion that goes into the lumen of their airways. This is actually the leading cause of death due to asthma, is plugging of the airways due to overabundance of mucus production. They also get big changes within the tissue itself. You can see thickening of the basement membrane, lots of collagen in deposition, and the muscle cells hypertrophy and hyperplasia. And so really these patients have very difficult times breathing. So the lab that I joined was very interested in this Th2 cell, and in particular, the control of this Th2 cell by RNAs. And the types of RNAs that we studied, and this was sort of what my first project in the lab was to study microRNAs. And hopefully some of you have heard of these. They're short non-coding RNAs. They're only 21 to 22 nucleotides in size, very small. They're transcribed in the nucleus, and they undergo a sequential set of processing until eventually they serve as post-transcriptional inhibitors of gene expression. And classically, they will bind to the 3' UTR of other mRNA transcripts, and they bind by imperfect base pairing. And what they do is they shut off gene expression. So it's another level of regulation of gene expression. So my first project as a postdoc was to look at a cluster of microRNAs and their role in Th2 cells in asthma, which was very interesting. I could talk about that, but I'm not going to. So I was finishing up that project, and we were kind of trying to decide what to do now, what to do next. And there was this emerging literature that showed that RNAs could be secreted into the extracellular space, although it wasn't entirely clear like what they might be doing there. And there wasn't much data on asthma. And so we began to think about whether we could find some inflammatory extracellular microRNAs in the allergic lung, actually in the extracellular space in fluids. And to do this, we started by collecting different biofluids, either in control mice, mice that were immunized, or mice that were immunized and then challenged in their lungs. So these are our allergic mice. We collected a lot of different samples, but today I'm just talking about this BAL fluid sample, which is the liquid basically that's washed up out of the lung. And when we sequenced that liquid in the lung, we can see that most microRNAs don't really change much. So this is a plot that shows you the overall expression level on the x-axis versus the change in expression on the y-axis. But there were a group of microRNAs that were very increased in our allergic lung. And there were two things that got us excited about this result. First of all, from kind of knowing the microRNA literature, we knew that many of these microRNAs, like miR-223, 142, 150, 363, all had known important functions in immune cells and often were highly expressed in immune cells. So we thought that maybe we were seeing this sort of immune cell signature in the lung fluid. The other thing that was really exciting is that there was one human study looking at sputum from patients with severe asthma, and they have upregulations of miR-223 and 142 as well. So this really suggested some physiologic relevance to what we were observing. Now, I'm not showing quite a bit of data about further characterization of these populations, but we thought that these were coming from immune cells. But correlation is not causation, and that's an important principle in science. And ideally, we would have really liked to show that these microRNAs were coming from immune cells. And I'm still working on that to this day, because that has been a very tough nut to crack. So I think sometimes in science, when you run up against a roadblock, at least a temporary one, it's good to think a bit laterally. And that's what we did. So we said, gosh, it is really hard to trace the source of RNAs from in vivo, but maybe we can trace something else as a proxy. And the thing that we got most interested in was that these RNAs typically are not just floating free in fluids, but they're actually encased within vesicles. And there's lots of different flavors of vesicles. There's exosomes and microvesicles. Some people call them small and large, EVs. But the point being that these RNAs were not existing in a vacuum, but seemed to be encased in a lipid structure. And we thought, well, we can't easily trace the RNAs, but maybe we can trace these vesicles. And so we developed a cell type specific membrane reporter that could help us to trace the origin of extracellular vesicles by flow cytometry. And this is kind of a complicated slide, but the important point were that we could basically in a tissue specific fashion, remove a membrane bound tomato and change it for a membrane-bound GFP. So this means in the mouse, we have a subset of cells that are green and a subset that are red. And we hypothesized that if these are tagging membranes, some of this should make it out into the vesicles that are secreted into the extracellular fluid. And if we see red and green vesicles, this would allow us to backtrace the origin of these vesicles. And we did that by EV flow cytometry. So we set up some mouse crosses and we made them. We made two mice, the MTMG NKX mouse, which labels epithelial cell green. So here I think you can see a bronchiole and this nice epithelium labeled green. And then a second promoter where we could label all immune cells. So no longer is this epithelium green, but you can see that there are little cells coming out of this blood vessel and lining this bronchiole that are green. So these are the immune cells in the space. And we did our vesicle flow cytometry. And so on this left-hand side, you can see membrane-bound tomato labeling, and on the right side, you see GFP. So what you can see is that most of the vesicles we can detect that are fluorescent positive are coming from the epithelium, but there are definitely a few that are coming from immune cells. And this just is in a control mouse. So about 80% are coming from epithelial cells. But what was really exciting is that when we induced inflammation in these mice, we saw an increase in the number of vesicles coming from immune cells, suggesting that as part of a tissue inflammatory response, immune cells are coming in, they're spitting out vesicles, and we think spitting out RNA. We still haven't directly proven that, but now we have this nice additional correlate and some additional, and this was really, I think, one of the first demonstrations of finding tissue specific extracellular vesicle secretion into the extracellular space. So this kind of has opened this whole new world as I've started my own lab to think about really what are the implications of packaging these RNAs into vesicles. And there really are just no end of questions. This is a young field, so it's a very wide open space. But we may think about cellular sources, the regulation of their export, RNA containing subsets, target cells, their function, therapeutic implications. So it's very exciting, I think, to be doing. So that is sort of what I did. Hopefully, yes, I did okay on my time. And I'm happy to take any questions.
Wow, thank you so much, Heather. Gosh, your passion for science is so clear, so transparent. I just appreciate that so much. I also studied gene regulation in grad school, so that was fun. I'm going to open this up for any questions that anybody might have. I have one that I'll start with. So you started with an interest in science and then gained an interest in medicine. How do those relate now? Could you have a lot of your time as science with clinic? Are you glad to have that clinic time? And yeah.
Yeah, I mean, I still am really glad to have that. And I think that it has changed the way I think about science a bit. And I also get a lot of personal satisfaction out of doing my clinical work. I think that for me, my clinical work is, I get to see the execution of science in an effective way. And in lab, we spend a lot of times doing things that don't work, which is fine. And actually, my lab thrives on that because we like to sit right at the frontier of the field. But definitely for me, it's really satisfying for me to go sign out cases where I know that scientific discoveries and assays that are being PCR assays or other types of assays, sequencing assays that are done every day in that lab result in a change in patient care. So that's really satisfying. And I think also it's opened some other interesting doors and avenues in terms of collaborations that I have. So we have really interesting collaborations to try to couple the sequencing of patients with severe inflammatory and autoimmune diseases with clinical care of those patients from pediatricians and folks in medicine and basic scientists trying to ask fundamental questions about how the immune system works. And it's a lot of fun to be a part, just one, small cog in that sort of network of people that I can offer actually the possibility of taking care of those patients. So right now we're actually validating tests to do for those patients here at Vanderbilt so that we can enhance that collaboration and really try to push forward our care of those patients and the science as well.
Wonderful. Thank you so much. I see a question here in the chat. Are patient donations or participation, is that part of the research you do? Do you actually use patient samples?
Yeah, so up to this point, I have been pretty much exclusively a mouse immunologist, so working really in basic systems. But over the last year or two, we've have a couple of collaborations with folks, especially in pulmonology, where we're starting to actually work with primary biofluids to look for these extracellular vesicles in allergic conditions. And so I sort of see that as an opportunity for growth for me over the course of my own career, coming from a pretty basic science background. I think, there's a huge spectrum in physician scientists. I will well acknowledge that I'm on that basic side of the spectrum, but it is still fun to sort of play around on that.
Wonderful. All right. Well, we hit the time perfectly. Thank you so much, Heather, for being here. It was such an honor to hear about your story. And gosh, we're just lucky to have you at Vanderbilt. Thank you so much for being here to talk to these students too.
Great. Thanks, everyone.
All right, everybody, I hope you have a wonderful weekend, and we'll see you next week to talk about admissions. Have a good one. Bye.
Check out our website to find recordings of all of our videos. We'll get them posted about a week after each of the presentations. We can't wait to see you!